Hello and welcome to week 15, Biotechnology. So this week we're going to be studying bioinformatics, which is the combination of biology and computer science, which is looking at big data to help understand biology and human genome. So we need to remember how big the human genome is and how many nucleotides are actually encoded in our 23 pairs of chromosomes. So we're going to be talking about big data, and the big part is just how vast our genome is. So some of these slides you've seen before, but the point of this is just to understand how many nucleotides, how much DNA is included in every single one of our cells. So when talking about the tools we're about to learn about, it's important to understand why we need these different tools. So this slide is a great example when we talk about big data, just how big we mean, and how you can see that there's been this overlap between computer technology and computer data storage and the type of questions we can ask biologically. We've got these huge genomic databases from countries all across the world collaborating for years and used as a resource for different scientists to use that have these large databases of genomic information. Here we can see a timeline of the key events in biotechnology and our ability to manipulate our genome and improve human health. So we started, and we already covered this pretty well in depth, the structure of DNA and that's discovery of that famous double helix. So that was in the early 1950s. By the early 1960s, we as humans had discovered the genetic code and that Rosetta Stone of how those nucleotide sequences get converted into protein. It took us about to the 70s to have this recombinant DNA technology I'm going to talk about in this presentation. And it's really been only within the last 40 years or being able to sequence our DNA and other species' DNA, manipulate that DNA in a process called PCR, you're going to learn about in a moment, and to really have the actual data for what is in the human genome. Biotechnology, we discovered DNA, now we need the tools to manipulate it. So restrictions enzymes, you can picture, are particular proteins that act like giant scissors. And so this is absolutely key because if you're picturing those millions and millions of nucleotides, if we want to be able to manipulate the genome, we need to be able to physically move it around to cut and paste it. So restriction enzymes were one of the first key tools that we had in our toolkit to be able to modify nucleotides. And essentially, again, they're like scissors that are going to cut at a particular sequence. So there's different enzymes, and this is, as we're going to see, the parallels with what we talked about with CRISPR-Cas9, as this is a part of a bacterial antiviral system for bacteria to be able to cut up that genetic sequence that viruses are injecting in to try to make the bacteria sick. So the bacteria have these tools, they have these scissors that can cut the DNA at different sequences. So why this is so key is because restriction enzymes allow you to cut DNA at a particular sequence, which is just this repeating nucleotides that might be multiple times within a particular chromosome, but you can cut one fragment and you can cut another fragment. And when that cutting is done with sticky ends, is what we call here, when they're not cut in one blunt slice straight through, but rather they leave overhangs of nucleotides, it allows different DNA fragments to come together, and using DNA ligase that we learned about from DNA replication, you can then link those two back together again. So what restriction enzymes do is they allow us to make recombinant DNA molecules. Recombinant DNA molecules is just that combination of different DNA from more than one organism, where you can use a restriction enzyme and cut, producing an overhang, and then combine different fragments of DNA together to make that recombinant DNA. So what is a key step in that recombinant DNA? is using those restriction enzymes at particular locations that you're going to be able to figure out what those overhangs are. Using the same enzyme, cut your gene of interest, cut your DNA sequence, and then you can glue it back together with ligase binding where those holes had been. So a cloning vector is simply just a stretch of DNA that can accept foreign DNA. So a plasmid are these circular bits of DNA that are in addition to the DNA which is in a bacterial chromosome. So these are small circular bits of DNA that are outside of a bacterial chromosome, and we as humans have manipulated this characteristic of bacteria and how they spread variation, not by sexual reproduction, but by swapping plasmids from one bacteria to another. And we have now made our own laboratory plasmids that have particular sites. They have particular different restriction enzymes. So each of these is a different type of restriction enzyme with particular sequence that you can cut open a plasmid with and insert the bit of DNA that you are interested in into that plasmid, and that plasmid can get uptake by a bacteria, and then that bacteria will forevermore contain those plasmids and be able to express the genes that are located within that chromosome fragment that you inserted into that special plasmid. Now, that special plasmid has other genes, in this case ampicillin or kanamycin, that are selective genes that can overcome antibiotics that are put into the media so that you can select for just the bacteria that have that bit of foreign DNA and are expressing that recombinant plasmid. So as we've been reading about in our discussions and in this week's assignment, CRISPR-Cas9 technology is this brand new technique that allows us to manipulate a genome very easily. And I cannot stress enough how revolutionary this technology is in allowing us to be incredibly specific for where in a genome we want to manipulate. So restriction enzymes, those are going to find any particular sequence that's just a few nucleotides long so that repeats throughout the genome. The CRISPR technology uses a guide RNA that evolved from this immune response from bacteria cells that are cutting parts of infecting virus and keeping part of that genome within the bacteria genome to be able to recognize that nucleotide sequence, and using this CRISPR-Cas9, I'll say technology, but it's the evolution within that bacterial cell, to then be able to cut up any recurring infection from that same bacteriophage. So we are using this system to manipulate genomes with incredible precision that had never occurred previously with any other techniques. So since we've had all of the discussions, I won't actually discuss this technology more here. What I will discuss is PCR, which is the polymerase chain reaction. This was actually one of many Nobel Prizes awarded to the tools that we've been describing, including restriction enzymes. But this was actually a Nobel Prize that was awarded for discovering a technique. And what polymerase chain reaction does is it allows us to amplify a particular section of DNA. It allows us to amplify DNA by using what are called primers that we can design that are going to go on the outside, are going to be just before and just after the sequence in question, and are going to have complementary base pairing so that in PCR, when you warm it up, the double helix is going to open up and separate, and then the primers are going to come down and bind, and then you're going to add a particular polymerase called Taq polymerase, which is able to operate at very hot temperatures, and that's going to fill in the rest of the sequence. And what PCR does is it does this for approximately 30 rounds, and at the end of the day, it results in the production of that particular sequence of interest at large quantities. So if you remember last week, I talked about needing to genotype mice that I needed for my double mutant, where we had to do the dihybrid cross, and asked, how do we genotype mice? Well, the first thing we're going to need to do is take a little bit of their DNA and do PCR. So you are going to be expected to understand how to do PCR, which is what's covered in Lab 1, which is what I was just explaining. That involves DNA polymerase, nucleotides, and primer. And it involves these series of heating and cooling cycles where you're opening up the DNA, primers bind at that lower temperature, you increase the temperature for Taq polymerase to go, and then you start the whole cycle over again. This is what's done in Lab 1. Here is actually from my lab. This was our actual protocol where you take a little bit of the tail of the mouse to get some of the DNA. You vortex it, so you mix it all up, and then we do PCR. And then we're going to separate that DNA product by gel electrophoresis. So to genotype, you can look for particular genes of interest, and you can add the different requirements for the PCR, the primers, the water, the actual cells, and then you have the PCR setup where you're cycling between these different temperatures for different amounts of time. So in the end, you produce your DNA fragment, and then you run it through gel in what we call gel electrophoresis. Now this we again saw in the lab where you are separating DNA fragments based on their length. Based on their length, because the smaller the fragment, the faster it moves. And the DNA fragments are moving based on their negative charges moving towards the positive end of a gel electrophoresis setup. Again, you will see this in your lab. So you can then place your DNA fragments into a well of an agarose gel. You apply the current, and those fragments will move. And based on the size of those fragments, you can determine what genes are present. So here you can see is the product of my genotyping back from my days in the lab. All the mice had the genes of interest. So the next technique is DNA sequencing. Now, sequencing was just revealing the order of the nucleotides. Now, the technology in order to do sequencing has dramatically increased. So it used to be someone's entire PhD thesis would be to sequence one gene, looking at doing gel electrophoresis and a technique that involved different nucleotides where you could be able to see what nucleotide was added in a one-by-one process. This was incredibly painstaking and time-consuming. The Human Genome Project began in 1990, so before I'm sure many of you were born, but it was really a turning point for biology when the sequence was first published in the year 2000. This first case where we actually could identify how many genes were in a human. And the number was actually far less than what we had been projecting. And this was really the beginning of where we see this watershed of sequencing and being able to identify the genome of now really any species. So the first genome cost $3 billion to get. And then what's been happening ever since was that price has gone down and down and down as our technology has improved. So that now we've got companies called 23andMe that aren't offering entire genome sequencing, but are looking at a fair bit of the genome, and you can do this for less than $100. So the tools and the techniques that we're able to do and how we can use big data is directly related to the cost of what we can study. So back to this little timeline, we talked about, the 50s, we got the structure of DNA, the 60s, understanding how it's working, starting to get these recombinant techniques in the 70s, and with this tool of PCR, and really putting it all together throughout the 90s. So what we've been talking about is this study of genomics, the study of genomes, and how we can use all this data from sequencing to understand what different genes are doing and how bodies and how life is operating. So one of the key things we can do with a lot of sequence data is compare different genomes. And we can use that to identify the evolutionary path that we're going to be talking about next week for how organisms are related from each other. We can look at the more similar the genomes are, the more closely they are related. And to give you some idea at the scale we're talking about for just how much information we are getting, when part of this presentation was made by another faculty in 2013, they had 189 different species had been sequenced. Most recently, when I checked, we're now over 1,300 different species. Yes, most of them are prokaryotes, but it's really unbelievable to think about how many species have been sequenced and how easily accessible this data is, and how quickly these transformations are occurring. So in our second lab, we looked at DNA profiling. I'm not going to spend too much time because, again, we focused on this in the lab, but looking at these short tandem repeats, which are small sections of DNA that are repeated for different amounts of time. So there's different lengths of fragments, and when you look at many different of these locations throughout the genome, you can identify if DNA came from the same individual based on having fragments of the same size. And so the lab you'll be looking at elephants. So another huge umbrella for terms of things we've been talking about and applications of biology is genetic engineering, where you are deliberately modifying an individual's genome. And this is, again, what we've been focusing on in those CRISPR discussions. But most people have actually heard of genetically modified organisms, or GMOs, as they're related to foods. And it's really important to understand how the food that we eat came to be from conventional breeding, which took wild type plants, in this case, but also animals, and bred the ones together that had the beneficial genes, that had the beneficial traits, and continue to cross them until you can continue to develop the either plant or animal that has the characteristics that you want. So that it's slow and costly, but as we see from just the foods that we eat, most of the plants, the fruits and the vegetables in particular, you can get these fantastic pictures of seeing just how much we as humans have been manipulating the genomes of the animals and plants that we interact with and eat since the dawn of agriculture. That's what agriculture is. It's just doing it at the level of plant breeding. Whereas now, with genetically modified organisms, it's not random backcrossing, but it's taking a gene from one organism, and in this method, showing through a plasmid, inserting it into a new plant. You can do this from organisms that are the same species, taking a gene and inserting it, or you can do a gene from a different species. That's transgenetics. This certainly produces that trait in a new plant much faster than the old mechanism of just breeding constantly. Now, genetically modified organisms are used in many, many different fields that you might not even think about. Ones that are particularly relevant for me this week, because we're going to be buying some glowfish, are fun things that aren't actually to improve medicine or human health. In this case, these are fish that are expressing fluorescent proteins, but more importantly are bacteria that are expressing human proteins and making things like human insulin in bacteria for diabetics. We've also been making designer plants as an application for this technology. There's some different ways to get DNA into a cell. Some of the most famous examples of genetically modified plants are BT corn, which makes the corn express an insecticide, essentially, a particular salt that will make it lethal to an insect who's eating it, but is harmless to humans. So that's an insecticide. There's also plants like rice that has now been made to express beta carotene that are making the plants healthier, that are making the plants be able to express certain proteins or vitamins that are not naturally occurring and can significantly improve human health. Lastly would be the famous or possibly infamous example of the herbicide glyphosate, which you might have heard more commonly as Roundup. And what this is that plants that have been genetically modified to express essentially a scissors or something that makes that specific plant resistant to the herbicide that is applied, so that fields can all be sprayed with the herbicide, which would normally kill all the plants, but the only ones that will survive then are the plants that are expressing this particular essentially scissors, and so that they are not killed by the application of the herbicide. So you always need to continue to buy the seeds that are going to be resistant to that Roundup. So again, all of this genetic engineering is producing organisms that would not be able to be made through just normal mating. It's really quite astounding to see how this technology is end up being used in research, in mice, in industry, to produce things to improve human health and human understanding. So one of the cases that's a particularly exciting lead of this genetically modifying organisms are to actually produce organs that could be transplanted into humans. I won't get into it too much, but the human immune system is really good at detecting things that are foreign. And so taking an organ from another human needs to take chronic medication in order to not reject their transplant. One of the grand desires of genetic engineering is to produce animals that could be the source of organs for humans. So lastly, sort of 1,000-foot level, we can talk overall about gene therapy. So gene therapy, again, transfers a normal or a modified gene into an individual with the goal of treating a defect or a disorder. Previously, there had been many issues with this, and I'm going to go through 2 examples and thinking about the history. So we used to have to do this with ways of using viruses in order to infect. Again, now, as we've been learning, CRISPR is fundamentally changing how we can do these things, which is critical because we used to have really unpredictable outcomes. So we've got a really wonderful story of an example of a boy who previously did not have an immune system, essentially. He essentially had that example of the little r from the mice that I was growing, from the mice that I was trying to create with the double mutants. Essentially, he had that genetic defect from birth, and so he did not have a strong immune system. What was done was he was able to have his cells removed from his body and using viruses insert the genes that were missing into those cells, where those cells were able to uptake that gene and start expressing and then multiply and actually return those specific cells to reconstitute his immune system. And this is one of those wonderful cases where you can see that he is now a healthy boy. A much more tragic example, which fundamentally set back genetic engineering for a significant amount of time, came from the fact that this boy, Jesse Gessinger, had a disease, had a genetic disorder, but it wasn't a lethal disease. It wasn't killing him. He had to eat a particularly restricted diet, and he certainly was not pleased with his disorder. It wasn't a fatal disorder. That he got a trial viral vector, which ended up activating his immune response, and he ended up dying shortly after the genetic therapy. And this really, really frightened the field and really made everything step back from a number of years. So now going to this slide of this timeline, thinking about sort of the most recent technology advances in biotechnology, we get up through thinking about DNA, how we can manipulate DNA, how we have this sequence of DNA to know what to be manipulating, and now we have this new tool, CRISPR. What I also added to this timeline over here at the very end is COVID-19. We're going to end this section on biotechnology to talk a little bit about how the COVID pandemic relates to the material we've been learning about. But I'm actually going to do that in a new video, so check the next video. Have a good one.